Okay, we are back again in a TYBSc Zoology Mammalian Histology paper number 2. We were discussing about the histology of kidney. We have seen the different structures in a gross section of a gross LS sections of kidney. In that now we are focused to the basic functional unit of kidney, that is a nephron. Now here we are looking at slide sections, color sections. In previous slide section, we have seen a different glomerulus, placing of glomerulus or nephron portions in cortex and medulla. Here now, this slide is more focused to in a cortex portions of this kidney. So here we will try to train our eyes to see the different parts of this nephron. So this portion is a glomerulus, and if you try to trace here, so all these are the tubes, convoluted tubes cut into different plane. So this is a proximal convoluted tubule. Then this lining indicate the distal convoluted tubules, and these are again renal corpuscle. Now, why we see these different textures in a proximal convoluted tubule? Here you see lot of pink color, a large lot of pink color structure is there, and here we see a less number of pink cytoplasmic structures and more nuclear structures here. Again, in this part, we see a very crumble structure. You cannot separate it as a. It is not a. We cannot see it here as a tube, but it is a very much mixed structure. Now, why we are saying that this is a corpuscle? In further few slides, I will acquaint you with the three-dimensional structure of a glomerulus. That again thereafter will revise the same slide, so you will able to imagine how it looks in a section. Now, why this? Question is why this uh, take arrangement of a convoluted tubule and distal convoluted tubule is different. Now see here, because of this epithelial cell type is different in this proximal convoluted tubule and distal convoluted tubule. So we see these differentiations and we able to identify that this is the distal convoluted, this is proximal convoluted tubule. Okay, this is PCT and this is DCT. All these DCT we can recognize very quickly. That is because of their different cell type. Now see what kind of cell we see in is a all internally all these tubules are lined with the basic epithelial cell based on a basal lamella. There is a base at the base, that is on the surrounding here. There is a basal lamella is there, a basement membrane is there over which this epithelial layer, cells layer is situated. Now, what kind of epithelial cell you will see in a proximal convoluted tubule from the glomerulus to the end, up to the uterus, up to the ureter, you will find everywhere there is a internal cell lining of epithelial tissues, but they change, their structure change. So in proximal convoluted tubule, you will find these are, you can say it's a cuboidal epithelial cells, but they have high amount of brush border. That means they are not cilia. A cilia-like extension of a border, and that is here it is called as a brush border. That is only the sole purpose is to increase the surface area, absorb the surface area. Okay, but in case of this distal convoluted tubule, they are again cuboidal epithelial cells with large prominent nucleus, but their brush border are very much reduced. Only one or two extension of this border are there. So you see here less amount of cytoplasm. Here more amount of cytoplasm is prominent with because of that increased brush border structure in an epithelial layer. Now this is a diagrammatic presentation of a nephron, a model, a three-dimensional, you can say the two-dimensional model to give you a perfect idea how that nephron is there in a kidney. Now, if you separate this region, so from here, this forward region is a cortex portion of a kidney, and this is a medulla portion of a kidney. So if you cut this kidney in this plane, in this straight line, what you will see a round structure of this tubule. Then here you will see a fraction cut of this tubule. And here is the round structure of tubule. If you cut this section in this way, you will find again these are cut in a different plane. So basically, we'll see this first. We'll see the different part of this nephron. Hmm. So it start from the glomerulus, a dense, dense mm, clump of a capillary network. It's covered with the cellular portion. That cup-like cellular covering over the capillary is called as a Bowman capsules, and this clump of a capillary is called as a glomerulus. And this is the actual site where the blood get filtered and they separate the waste product here and they retain WBC and RBC in a blood. So except WBC and RBC, everything get filtered into the Bowman capsules, and essential things are then get reabsorbed while passing through this tubule system, and unwanted things are poured into the collecting tubule, and that is what we call as a filtration function of a filtration. So not only these glomerulus is blood vessels are not only involved only in these glomerulus portions, but all these tubules are highly circulated, highly covered the network of capillaries you will find surrounding all these tubules also, because when we say it's reabsorption take place, reabsorption of useful material take place in this tubule system, that time for reabsorption it required the surrounding network of a blood capillary. In some slide we'll see that network. So basically this is a glomerulus, this is a Bowman capsule. They have funnel-like structures and they are connected with these tubules with the neck portion here. And so these Bowman capsules lumen is connected with this neck portion, and that starts the proximal convoluted tubule. Now to remember this proximal word, this is previous previous part of a nephron structure, that is proximal, and distal means far away from the neph these Bowman capsules, so their name is a distal converted tubule. Okay, now here you see that texture of a particular cells over these tubules. So all these cells are tissues here forming this tubule system is basically they are epithelial tissues. In proximal convoluted tubules, all they are cuboidal cells with large amount of a brush border. Thereafter, in this descending loop of Henle. Now, this is terminology descending and ascending loop of Henle, and these are the thick segments here. Okay, so here when that proximal convoluted tubule change into the descending limb of Henle, that time the cellular type changes from cuboidal to the squamous, but still they have large nucleus and little bit brush border you will see in a epithelial cells of this descending tubule. And when the same cellular type reaches to the loop of Henle, they are very much flat and totally squamous epithelial tissue. Here there is no cuboidal appearances, there are no brush border appearances there. It's just a um, thin squamous epithelial tissue as we seen in a that capillary blood capillaries. Okay, then as it reunite or they enlarge here in a ascending loop of Henle, they again form a structure, epithelial structure as a cuboidal structure, and here again it start producing little bit brush border, but that is as compared to this distal and proximal tubule that they are number of brush border are very low. Now, when this tube again forward as a distal converter tubule, there you will find uh, the cell type is again the cuboidal, but surface area is flat with lit- uh, almost no brush border. Okay, so this collecting tubule again, when continue, the cellular type is a squamous epithelial tissue lining is there in a collecting tubule. Now, overall, here you will find a very flat squamous epithelial tissue. No absorption take place here. It just tissues are there just to form a structure, Norman capsule. Then from the neck portion, they start the cuboidal epithelial layer. It little bit reduce here in a descending loop of Henle, or you can say the thick segment, and in thin segment it is a flat squamous epithelium. Again, in a this ascending loop of Henle, you will find it is large cuboidal epithelium. Here it is again the cuboidal epithelium in a distal convoluted tubule, and in a collecting duct it is a flat squamous epithelium. Now here, just for reference purpose, as we are talking about the filtration and kidney, the filtration unit, all thing absorption, reabsorption kind of a word. So we see a nephron structure, but we don't see a blood supply. We don't discuss blood supply anytime. So here, when we say reabsorption, it is not only a function of tubules to reabsorb, but they have to pour that reabsorbed materials into the blood supply. So all these tubules are highly supplied with a network of capillaries. So we'll see here the some of a label of some of these blood supply arteries and veins. So here the major artery and veins. In a major artery which enter into the kidney is the renal artery. Then it goes on branching. It forms a interlobular arteries and same labeling with the vein, interlobular veins. Then when they reach into the cortex portion, they are called as a arcuate artery and arcuate veins. Then again they bifurcate and they spread their branches into the very far cortex portion, that is its veins and artery, then called as the interlobular artery and veins. Okay. So this is how they produce the dense network of blood vessels in a kidney to facilitate the reabsorption and secretion portions in a kidney. Majorly all reabsorption part take place through this network of arteries and veins, and only the pouring portion, that is um, filtration portion, take place in a glomerulus. Now here again you can see how these blood vessels densely forms network around the nephron. So if we start stressing here, this is a glomerulus. So the blood vessels pouring blood into the glomerulus, then see these branches of these arteries and veins surrounding these tubules. So everything what required to be absorbed, it get absorbed back into the capillary network here, and it may be either pour back into the glomerulus, or it is, if it is not filterable, and it is reabsorbed back into the blood system, vascular system. Now see this dark color line is a collecting tubule, and all these blue color lines indicate the veins, and red color lines indicate the blood supply. So basically the point is all that nephrons are highly surrounded with a network of blood vessels for reabsorption. Okay. Now here you will get the idea, perfect idea of what is that reabsorption thing is. Okay, vascularization of a nephron. So blood circulatory system present, or blood vessel system present around the nephron is shown in this diagram. So here this is a glomerulus, that afferent arteriole. Now this word afferent and efferent arteriole. These two words, if you able to remember, that will help you in writing or discussing the even the physiology portion of a excretory system. So when interlobular artery produces a branch and that branch goes into the glomerulus, that growing artery is called as a afferent and leaving artery is called as a efferent. The one which comes out of a glomerulus, that is efferent, and the one which goes inside the Bowman capsule forming that glomerulus, that is called as the afferent. Okay. So this afferent arteriole then further goes forward and goes on encircling all these proximal convoluted tubule, distal convoluted tubule, okay, and some branches they circulate all that thin and thin segment of this Henle's loop. So basic idea, physiological idea is whatever they lose from the blood through this glomerulus, that all essential things are get reabsorbed here, and filtration take place in this cuboidal epithelial layer. Cuboidal and squamous epithelial layer are responsible for actual filtration. Not here they don't do any filtration work. Here they just do a pouring work. Except RBC and WBC, everything present in blood is poured into these Bowman capsules, and real filtration takes place at the epithelial layer present in this distal and proximal convoluted tubule. That cuboidal cells, epithelial cells, the brush border thing, all these are they play role in a actual filtration where they reabsorb the essential thing and pour back into this capillary. Then these capillary unite together and they form this vein, and the useful material is then again poured into the main bloodstream through these veins. Okay, so I hope you will understood this network system. This vasa recta word you may found in some of these next slides. So this vasa recta is what is the network of blood capillary form around the thin segment of Henle's loop. That is called as a vasa recta, because while observing the sections you will find lot many blood vessels cut in a sections. That blood vessels are of a vasa recta. Okay, this is a model of a glomerulus to help you imagine what that glomerulus. So this is a incoming artery, that is afferent artery, and this is outgoing artery from the glomerulus. You can see the incoming arterioles and afferent arteriole and efferent arteriole. Now, and once they enter in a glomerulus, they again use branches of small capillaries here. Now, the main point in this capillary is they are of fenestrated type of capillary. You remember that fenestrated word what we discussed while seeing that capillary histology. Fenestrated means they have very large pores. They don't control much filtrations. They have very large pores, so fenestrations word is used. So maximum thing get removed here. Along with this, in the capillary structure, these capillaries are provided with these podocytes, specialized cells which you find only in a glomerulus, which control the opening and closing of these fenestrated capillary. The podocytes, they guard that opening. Okay, so rest all internal lining of this Bowman capsule is lined with the squamous epithelial tissue. Even here you can see the squamous epithelial tissue. As the neck start forming, it shows increased size, that is a cuboidal structure here. Okay, now when you inject a particular dye in a kidney which stain blood present in a kidney, that gives such kind of a appearance. So here the cortex, when the dye, inject that dye in a cortex portion, it reveals the vasculatures. So all these brown color portions are blood vessels. So this just to identify the glomerulus. The blood present in a glomerulus gets stained, and you see these are the all glomerulus. Okay, and these are the major interlobular arteries and vessels, the blood vessels are there, then accurate vessels and peritubular capillary network. Okay. So all these indicate the presence of glomeruli that help us to identify the glomeruli. And all these yellow color texture portion is a tubule system. Okay, so in exam or in your notebook, you are supposed to draw this diagrammatic drawing of structure of renal corpuscle. Renal corpuscle means a group of the part of nephron which comprises Bowman capsules and glomerulus together. So that is the renal corpuscle, the single unit of that functional unit of a kidney. Okay, so here in renal corpuscle, the components are some part of a neck portion of a Bowman capsule. This is the Bowman capsules, and these entering and leaving arterioles, and this one more part comes back again in contact with the glomerulus. In earlier few slide, I told you about that. It's a distal tubule. This distal convoluted tubule come back from far away from the medulla to touch to this glomerulus. And the significance is there they form a macula densa kind of a tissue which plays a role in secretion of some hormones which maintain the flow of urine, or they maintain the filtration rate, and is in terms they maintain the blood pressure and lot many things. Okay, so this connection zone where this distal convoluted tubule re come back to touch to this afferent arteriole near the glomerulus, that portion is called as a juxtagranular junction. This is again, remember, this is what when we say juxtagranular junctions or juxtagranular structure, this is what it is, a reunion of wall of distal tubule with the afferent arteriole. That portion is called as the juxtagranular portion, juxtagranular junction. Here, because of these connections, there forms this macula densa. We'll see the specialized function of this macula densa in next few slides. Then the cell types in junction of these two arteries and one tubule, that is a lattice cells, are specialized cells formed there. And this area of arterioles which come in contact with this distal tubule wall, that cells are called as the juxtagranular cells. Okay. Thereafter, in this Bowman capsule, we generally use this terminology as a vascular pole here and renal pole here. So where all filtrate except RBC and WBC goes into the tubule system, that area or that part of this Bowman capsule is called as a renal pole, and this area or opening through which the glomerulus or arteriole enter into the Bowman capsule is referred as a vascular pole. Okay. Then if you see this glomerulus of blood capillaries, what you will notice here is a mesogial cells scattered in the gaps between the capillaries, and these are glomerular capillaries and a podocyte, a visceral epithelial. These podocytes which cover this capillary, and they have the role in controlling the filtration rate. And this internal space is a capsular space. So in three-dimensionally, you will see this. These are the tubules, capillaries entered in, and they form the network. And this is a ball or a round structure, capsule-like structures forming, that is the capsules made up of a smooth squamous epithelial tissues.